After the construction of standard graph of glucose, so as we have already learned that it can be a graph of standard graph of glucose or standard graph of maltose, which are supposed to be the products formed as a result of amylase catalyzed reaction. So in this fashion, after the construction of this standard graph of glucose, now it is possible to convert optical density readings into glucose concentration readings. So the next step will be processing of the sample in the same fashion the way these dilutions are processed. We need to process the sample also. So this is the protocol for the sample or the protocol for the test. That means this is the sample in which you want to determine the amylase activity. So this is the supernatant as a result of production. This is centrifuge. The cells are settled down and now the supernatant is ready for the measurement. We want to determine what is the concentration of amylase in this given supernatant. So this is called as crude extract. For this, the protocol is like this. We need to take two tubes. One tube will be labeled as test. Another tube will be labeled as blank. Now you have to make additions in this fashion. So in the tube labeled as test, add 1% of starch solution, 0.9 ml. 0.9 ml of starch solution is also added to the tube of blank. Remember that starch is a substrate for amylase enzyme. Next step is addition of the sample. That means the preparation which is under assay. That is amylase preparation, the crude extract. So from that crude extract, it is appropriately diluted and 0.1 ml of that is added to this tube labeled as test. Amylase preparation is not going to be added in this tube labeled as blank. Then enzyme catalyzed reaction is allowed to proceed for 10 minutes at appropriate temperature. So here in this case the optimum temperature is 37 degrees Celsius. So this preparation is mixed. It is incubated at 37 degrees Celsius in a water bath for 10 minutes. After the enzyme catalyzed reaction, for example, the amylase has acted upon starch and that leads to the production of the products like glucose and maltose. So after that, we need to determine or detect how much glucose or maltose is synthesized as a result of degradation of starch, as a result of amylase catalyzed reaction on starch. So for that, again, we need to add this DNSA reagent. This DNSA reagent, we have used it earlier for the preparation of standard graph. So you need to process these samples the same way as we did in the earlier slides, which describes the production of or the preparation of standard graph. So here again we need to make addition of DNSA reagent. It is added in the quantity of 1 ml to test tube. Similarly, it is to be added in 1 ml quantity to blank, because we know that this DNSA reagent it is a coloring reagent. Then Amylase preparation, there is no addition in test because it has been earlier added. And then after that, in blank, we can add that amylase preparation. And then these tubes will be kept in boiling water bath for 10 minutes. Remember that here, even if we add amylase preparation, the reaction is not going to proceed because we have already added a DNase reagent and the tubes are being now exposed at 100 degrees Celsius for 10 minutes. So biological activities will be permanently deactivated, permanently stopped. After that, allow the containers to cool and then add 8 ml of distilled water in taste and blank. So this protocol is same protocol which we have followed for the preparation of standard graph. So this is for the detection of reducing sugar by DNSA method. And finally, read the absorbance of that sample test at 540 nanometers. So we'll go to graph, standard graph, and we'll put that value onto this graph. So this is a straight line. So whatever the value of test is there, optical density of test, you can convert, you can plot that onto this linear part, and you can easily convert that optical density reading into glucose. Or we can make use of this equation, y is equal to mx plus c. So it is very easy to convert optical density reading to the glucose concentration. So using this standard graph, now say for example, this is your sample and you have got this kind of color. So from graph, the reading is, say for example, 0.478, the optical density is 0.748. Then you go back to that graph and plot this reading on the linear part of the straight line, and this will, and find out the concentration of glucose corresponding to this reading. So from graph it comes to 2 millimoles of glucose. Then we can convert this millimoles into micromoles by multiplying it by 1000, because 1 millimole is equal to 1000 micromoles. So 2 into 1000, it will be 2000 micromoles of glucose. Then the definition of amylase activity, as we know, it is the concentration of the product micromoles, that is in micromoles of the product form per ml per minute. So these two factors are there, that is time factor is there and the quantity factor is also there. So and if the original preparation is diluted, then there will be multiplication by dilution factor. That means if the crude sample of amylase, if it is diluted 10 times, 20 times, 30 times, then there will be a multiplication factor of 10, 20 or 30 likewise. So now we'll find out the amylase activity. Now from graph, what we have calculated is the concentration of the product is 2000 micromoles. Now the quantity or the sample which we have taken is 0.1 ml. So amylase preparation was taken in 0.1 ml. You can go back and see the protocol for test. So it is 0.1 ml. You want to convert that into per ml. So 2000 will be divided by 0.1 and it will be 20,000 micromoles. Now this is 20,000 micromoles per ml. Now you want to convert this into per minute. Remember that in the protocol for test, the incubation period was 10 minutes. So for this is the amount of product formed after 10 minutes. Now you want to convert that into per minute. So 20,000 will be divided by 10 and the answer will be 2000 micromoles per ml per minute. This is the enzyme activity. So the enzyme activity is now 2000 micromoles per ml per minute. If now there is no dilution factor, so now we can consider that what is the total quantity. That means you have expressed the activity for 1 ml only, but the crude extract, that is called as the fraction volume, that may be 10 ml, 20 ml, 30 ml, 100 ml, 1000 ml or whatever. So if you want to determine total activity, so this is per ml activity, you can multiply that with this fraction volume. Say for example, you have 100 ml of that supernatant, that is the crude extract, then this 2000 into 100, it will be 200,000 units. So this is variable, it can be anything. This is how you determine the amylase activity or enzyme activity. Now we know that the next part we have to determine is determination of specific enzyme activity. Specific enzyme activity means the activity of that enzyme in the given preparation of protein. So you have a protein solution, it will be a mixture of proteins. Among them, your desirable enzyme is one of them, and you want to determine what is the activity of that amylase molecules present in that protein mixture preparation. That is called as specific activity. So for that original sample will process that original sample to determine total protein content in milligrams per ml. So this part is discussed or might have described earlier in another practical, that protein content we can determine by Follin-Lowry method. You can go back and check the Follin-Lowry method protocol, and using this Follin-Lowry method, the protein content of that crude preparation is 200 milligrams per ml. Now we know the formula for specific activity is enzyme activity, that is units per ml divided by total protein. So here in this case, the enzyme activity, if we consider it as 2000, total protein is 200, so 2000 divided by 200, it will be 10 units per milligram. This is the specific activity of amylase.